All right, welcome back to another lesson. This one's going to be about mutations. We've done all our talk we need to do about transcription and translation. Hopefully you've seen those lectures and have a good understanding already of how DNA is transcribed into messenger RNA and how that messenger RNA is translated to make an amino acid sequence, also known as a polypeptide, which will then be a protein. And so we're going to look at what happens when that sequence gets messed up. Now, technically, that sequence could get messed up anywhere along the line. You could, have a mutation that changes the sequence of RNA or DNA, but we're going to be looking at DNA mutations here. And so I think the book really watered this section down a little bit, so hopefully I can give you guys some freebie knowledge here. But let's look at what the book does have to say, is what you're accountable for. Literally, a mutation is simply that simple, and the fact that all it is a change in the nucleotide sequence of a molecule of DNA. And so, since DNA ultimately carries the code and is going to get rewritten as RNA, and then that RNA is going to get translated, well, if you have any mistakes here in the DNA, then you're going to get a different protein, right? First of all, how does that happen? Okay. And what happens in different cells? Well, first of all, if it occurs in a non-reproductive cell, which is basically every cell in your body, unless it's a sperm or an egg, then the mutation is passed on to daughter cells within the organism, but not necessarily transmitted to the next generation, obviously, right? So if you have a mutation in one of your skin cells and it replicates, well, guess what? That new skin cell is going to have the same mutation because it got DNA replication. It copied the same mutation. But if mutations occur in an egg or a sperm cell, guess what? The new baby, the new organism, that next generation is going to get that same mutation. It's going to get copied and copied and copied and copied. And so that organism is going to live with that mutation for all of their cells. But why? Why do mutations occur? That's a great question that scientists are actually still trying to answer. Now, some things we do know is that there are, again, just random events that we don't really can explain, but then there are some that are caused by mutagens. Those are chemical substances that we know will alter the structure of DNA. So a mutagen could be different chemicals that you ingest, that, like tobacco smoke, for example, is a huge one that's already been tested and proven years ago that tobacco smoke causes lung cancer because there's like thousands of chemicals, first of all, in tobacco smoke. People are just after the nicotine, but it's got all those other chemicals too. And those a lot of them thousands of them actually, are mutagens, and they mess up the DNA in your lungs, and so you end up with lung cancer. But other things too, like exposure to high radiation, like X-rays or even ultraviolet light from the sun, can cause mutations in your skin. So, but in general, what type of mutations do you have? And this, right? Sorry, just going back to this sentence. That was a short sentence, a short little paragraph. But again, so much is being explored right now as to what causes cancer or what causes mutations in DNA. And there's a lot of stuff out there, so we really need to be careful. But let's look at what type of mutations there are. So you have what's called a point mutation that literally just happens at a point. And those aren't always so bad. Like, for example, here's your original DNA. Well, what if we just replaced guanine with cytosine? Then we would have this new sequence with the point mutation there, right? Having cytosine instead of the guanine because we replaced G with C. Well, that may not have tremendous effects, or it could. It depends, right, on what protein ends up getting made. But a point mutation, the point of a point mutation is that it happens at a point. Okay, you're just affecting this one nucleotide. However, there are something called frame shift mutations, which, again, I don't like how the book didn't even mention that word. and there's chromosomal mutations that it didn't even talk about. So I'll show you a different slide here in a minute that might just help you understand this a little bit better, so you don't get this basic knowledge here. This is like super simple, and yeah, they've left some stuff out. But you have what's called a frame shift mutation, which shifts the whole frame. So how does that happen? Well, think about a deletion. If you did a deletion where you literally take out a nucleotide, so if we take out this G, instead of replace it with C, we just take it out. We just lose G. Well, what happens? Well, now your DNA is actually one nucleotide less. It's shorter, isn't it? But that has a profound effect because you are reading codons 3 bases at a time. So if you shift everything over one nucleotide, nothing makes sense anymore as the way it used to. Or vice versa, an insertion mutation occurs where, let's just say we just add a nucleotide. We don't take anything out, we don't replace anything, we just literally add a nucleotide like cytosine in here. Now it's one nucleotide longer. But again, what if we were reading this like GAG and TTC? Those were the first 2 codons. Well, now our first 2 codons are GAG, CTT, and then C, whatever. Like we could have kept going, but it literally shifts the whole frame from here on out. And so I'll show you a better picture again. Again, the book did a bad job of that. I'll show you a better picture of what a frame shift mutation looks like. But deletions and insertions will shift the whole frame. It's a frame shift mutation. Whereas a point mutation occurs where you just have substitution. Okay. And again, let's just look at the effects again. How the book lays them out here. A mutation can have negligible, literally minimal, or it could have catastrophic effects. And so to understand this, let's look at the following. Let's take the sequence CTT, and let's look at what happens when we do a mutation. So first of all, CTT is going to be rewritten as mRNA as GAA, right? And then GAA will be the codon, obviously, because that's mRNA. And that codon codes for glutamic acid. Okay? Let's say we replace this third T with a C. We do a point mutation. Well, now our new codon is GAG. But guess what? If you look at the codon wheel, or the codon translating wheel, the genetic code translator table, whatever you're looking at, you'll see that GAG also codes for glutamic acid. So we literally have a negligible effect. That mutation made no difference whatsoever. All right. But what if we take that mutation and we make the middle T? If we turn that one into an A, well, then what happens? Well, CAT will be rewritten as GUA in the mRNA transcript. And then after that's translated, it'll be translated for this will be the codon for the amino acid valine. Well, that's a different amino acid. Remember, amino acids have side chains that interact with each other and all that, and so that's eventually going to affect the shape of the protein. So you're going to have a totally different protein, possibly. Now, that actually depends on what protein you are, because there's actually some proteins that it really wouldn't matter. You could put valine there, or you could have glutamic acid. It wasn't really a major amino acid in the sequence to begin with. You know, certain amino acids have stronger attractions to other amino acids. We learned that in the last chapter. So let's say we had a non-polar amino acid here, and we put another non-polar amino acid there. Well, even though it's a different amino acid, they're both non-polar, so you're still going to get the same side chain interactions. But let's say you needed cysteine there, because if you remember, cysteine is the only one that does a disulfide bond. Well, if you put anything else there, you won't have that disulfide bond, which was the strongest of the 4 side chain interactions. And because remember, that's a covalent bond. A disulfide bond or a disulfide bridge is a covalent bond, and it's the strongest of the amino acid side chains. That always occurs between a cysteine and a cysteine. Well, let's say you have a cysteine on somewhere on the protein, but yet where the other cysteine was, now you have a non-polar. Let's go to the extreme. A non-polar amino acid that would not bond, then you'd have a major problem. So it depends. Now here's another time it would depend. Let's say you accidentally did this mutation where you had a point mutation where you throw in an A there. Instead of CTT, it's ATT. Well, that is going to be rewritten as UAA in the mRNA sequence, and that codon codes for stop. So you literally shut down the whole process. Like, what if that happened early on in the thousands of nucleotides that are there that end up needed to be translated? Well, you'd be stopping the process way too early. So stop codon would be bad in that spot. So, here's a sample problem. What dipeptide is produced from the following DNA? Okay, well, if there's the DNA, before we even make a peptide, we need to make a transcript. But let's finish this problem. B says, what happens to the dipeptide when a point mutation occurs and the DNA segment contains the sequence ATGAT? So we replace this G with a T. What will happen instead? So you got to do all the work. You got to turn this into a transcript, and then take that transcript and translate it into an amino acid sequence, and do the same thing for this sequence. And what you'll see is that after you write these two transcripts, they do code for different sequences. This would be ceruleucine, and then this one will be tyrosine and leucine. Okay, and that's two totally different dipeptides. And that could probably have a huge impact if it's just a dipeptide. If you have thousands of amino acids in a polypeptide, then just switching out one for another may not have a big a deal. But if you're just dealing with two amino acids anyway, you're just dealing with a dipeptide, like remember the enkephalins, or those weren't dipeptides, but they were small peptides. In the last chapter of proteins, we dealt with small peptides that were molecules like I can't remember the name off the top of my head, but we had a few. I think the enkephalins were the ones that I remember right away. They were like the painkillers. So swapping out those amino acids could have profound effects, because you're just dealing with a few anyway to begin with. So here's some problems to do of your own. And so there's where your genetic diseases come from, like cystic fibrosis, for example, is one of them I just read about here. This is kind of sad, but what you can see here is I did not know this was the treatment, and I've heard of cystic fibrosis, but I've never really known in depth what happens. It's a genetic disease, that's a wide open field. Genetic diseases, you could probably infinite amount of genetic diseases, and they're going to manifest different symptoms, obviously. But cystic fibrosis is when you have a mutation in for this protein here. It's called transmembrane cystic fibrosis transmembrane conductance regulator, and so that's needed for the proper passage of ions across cell membranes. Well, that's going to be a big deal, because pretty much every chemical reaction in the body is pretty dependent on the movement or the passage of ions across cell membranes. So that could have thousands of symptoms, I would think. But one of the ones that stands out with individuals that have cystic fibrosis that don't have that regulatory protein is they end up with thick mucus in their lungs, and it's got to be like pounded off. Like you can see this patient is receiving chest physiotherapy that involves pounding on the chest to dislodge the thick mucus that is clogging the lungs. Again, it has to do with ultimately the inability to move ions across cell membranes, but there's going to be other things too, like that individuals with cystic fibrosis are going to have decreased secretions of pancreatic enzymes, resulting in a failure to thrive or poor growth. And so there you go. There you have it. That's all they say about mutations. Just real quick, I did want to show you a pretty interesting picture here. So here, oh yeah, I wanted to show you this too. This is awesome. I was watching a video, the SciShow. He has a pretty good video about coronavirus. There's a whole bunch of them he's doing right now. The guy that does the crash course, Seth Green. And there's a West Coast and an East Coast coronavirus, interestingly enough. Now, there's a lot of caution out there as to how relevant this study really is. But the point is that on one of the lineages of the coronavirus, when they'd sequenced the genome, one of the amino acids is aspartic acid, which I don't know why it's abbreviated D, but it is. The other one is glutamic acid. So just that one mutation suggests, at least to certain biochemists, or whoever's doing the study, that there's like a West Coast virus, East strain or whatever, and an East Coast strain, because the coronavirus is, yes, it's mutating. Like, that's all. Viruses are constantly being mutated. One of the problems with the one right now that we're dealing with is it's mutated so severely that it's not like the common cold, and it's super infectious because it's mutated to the point where we don't have any like antibodies and we don't recognize it, and it's having terrible effects on us, and it's really contagious as well because of that. And there's so many factors. But anyways, the point I'm trying to make here is that one amino acid difference could be an indicator that this strain on the West Coast came from maybe someone on a cruise out here or something, and the strain on the East Coast may have came from someone that flying in from China or something like that, or from Europe. I'm sorry, because Europe is closer to the East Coast. But there's a lot of people that said that be careful with that study. Here's I wanted to show you this though. This is actually the genome for the coronavirus. 2,000, 4,000, 29,000 nucleotides long. And if you were to look at just this region, this gene here for the spike protein, if you're thinking about doing your coronavirus presentation, this might be something you might want to focus on. This gene here for the spike protein that goes from 22,000 to 5,000 nucleotide range. If you were to zoom in, that's what these dotted lines say. They say come down here. Let's zoom in a little bit closer. You can see that there's even sections along that gene where there's an S1 subunit and an S2 subunit, and there's even little sections there we can zoom in on, and we can see the actual sequence of DNA here. And they're just literally minor. I can't zoom in on this picture anymore, sorry, but there's just minor differences there between, let's say, like the bat SARS coronavirus and a pangolin coronavirus and a human SARS-CoV-2 coronavirus. And so what's fascinating to me is they've actually got all this stuff mapped out. So you can see there's so many similarities in the nucleotides, but then every now and then you have some differences, right? You have some major differences. And this would be a fun thing to look at and study. And if you have any free time on your hand, which I think you do, you could look at some of this stuff, and because now that you understand it, it should make sense. This is also what I wanted to show you. The different types of mutations. Here's a pretty cool way to say this. DNA has all you can ask for. Well, if you replace U with O, doesn't the sentence still make sense? So that would be what's called a silent point mutation. A missense point mutation when you end up with a word that doesn't even make sense. So that could be bad. Nonsense point mutations where you have an early stop codon. So then it's like DNA has all you, well, what? You never finished the sentence because you ran into a stop codon. Insertion mutations, those are frame shift. So if you put a Y there, look how everything changes because every codons are 3 letters at a time. So DNA has all you can ask, everything, but all the letters are still there. If you just took out this Y, the red Y, and moved everything back over, you'd have a complete sentence again that says DNA has all you can ask for. If you take a nucleotide out, you still end up with a frame shift mutation where, again, everything down the line now does not make sense because the first letter of this word is now the last letter of this word and so on. So everything got shifted over. And because DNA is read 3 nucleotides at a time, frame shift mutations are terrible. Insertion and deletion cause frame shift mutations. You could also have inversion where you just have a swap there like that. That would be bad. And then you can have translocation where you literally have a whole codon from one gets switched to another. Like you can have chromosomal mutations where parts of a whole chromosome get moved over to another chromosome. That can easily happen. Not always bad though, but so there you go. I'll probably just hold you accountable to the ones in the book though. So I just wanted you to see that there are lots of different mutations. And so I think we just got one more lesson about 17.10, focus on the human body, viruses, how relevant is that, and fingerprinting. I'll make that video now, and we'll be done. So thank you for watching. One more video left. Hang in there, guys.